Okay, I think we're going to go ahead and get started. It's just a little bit afternoon. So good afternoon to everyone. Thank you for joining us in our noon hour today. My name is Jan Hirsch, and I am the Dean of the School of Pharmacy and Pharmaceutical Sciences at University of California, Irvine. So welcome, everyone. We have put together a program for you. It's 1 hour. And we really are going to talk about just some basics about the vaccine. There's so many questions out there, and we thought it would be useful if we brought together people from within our school, because we actually have two departments. One is full of pharmaceutical scientists that are developing and discovering therapeutics and vaccines and tests, and the other side is pharmacists, our clinical pharmacy faculty, who are involved with patients and administering and distributing the vaccine. So we're bringing those two together for you today to answer some basic questions. So a little bit of housekeeping first. If you have questions, and we have received a number already from people that have signed up for this, if you have questions, please go to we're going to use the Q&A function, which you should see kind of at the bottom of your screen. And if you type in your question there, then we will do our best to get to it. We're going to have about 35 minutes of remarks from our three speakers, who I'll introduce in a moment. And then the remaining time will be for questions and answers. So next slide, please. So here we are. We have our speakers for today. As I said, I'm Jan Hirsch, the Dean of the School of Pharmacy and Pharmaceutical Sciences, and I'll be the moderator today. Then we also have Dr. Rob Spitali over on the left of this screen. He is a professor in our school, and he is our Associate Dean of Research. And he has a breadth of knowledge in mRNA research, which you might recognize because we have mRNA vaccines right now. And then next to speak will be Dr. Alex Chan. He is a professor in the Department of Clinical Pharmacy within our school, and he is also chair. And he is currently heavily involved actually in managing the pharmacy services at our Bren Center at UCI, where we are administering vaccinations. And then third, we have Dr. Carrie Hurley Kim. And she is a health sciences assistant clinical professor in our school also. And she also has, outside of that role, she's vice chair of the Immunization Coalition of Los Angeles County and a member of the LA County Department of Public Health COVID vaccine work group. She has a clinical practice in LA, although she works for us in Orange County also. So that explains that crossover. Next slide, please. So we're going to cover basics. Why are vaccines important? Basics about how the vaccines work, safety and efficacy of the vaccines, and distribution of the vaccines. Next slide, please. If you're following this as closely as many people are, you might be used to this, this little picture here. This is Orange County's tier rating. Right now, this was as of the 7th. Positive cases per 100,000, we're at 39. Those actually have decreased. We are actually decreasing our numbers. As of, I looked this morning, and I think that was more at 30. And the testing positivity rate was 10.9% on the 7th. It's now 9.4%. So we're moving down. We're going in a good direction, but we have a long way to go. Next slide. The other things that you might not look at as much are what's happening over time. That's where we are in one snapshot of time. Over on the left side, these are our daily cases. And the daily cases, definitely, we had a huge surge in January. Good news is those daily cases are declining. But we're still higher, that certainly than we were back in the fall. And get close to higher than we were even in the summer. So not out of the woods by any means. The graph on the bottom is for the hospitalizations and ICU patients. The top is the hospitalizations, and you can see the hospitalizations have come down, as have the ICU patients, and those are things that we look for in our tiering system. Next slide. And this is why we're here today. All right, this is an estimation, and you've heard a lot about models. So this is a model, and it's not true. It's a model of what could happen. And this is a model that's looking at the projected daily incidence of COVID-19 per 10,000 population. And the top curve in the black is without vaccination. And then the two curves underneath that are the sort of brownish one. The next one is with vaccination and with a reduction in a vaccine efficacy, because we've talked about that a little bit in other forums with the different types of vaccines, maybe, and variants possibly. And the light blue under that is with a vaccination and without a reduction in efficacy. And you can tell there's not much difference. And the point of me presenting this slide to you is because this is the why. This is the big difference that vaccinations can make. Right? That difference between the black line and those two lines below it, that's what we're trying to avoid with vaccinations. So just to set the stage for that. And now I'm going to turn it over. Can I have the next slide, please? Yes, and these are the two vaccines that we have right now. So we will start talking about those, the Pfizer and the Moderna. And now I'm going to turn it over to Dr. Spitali, who's going to give us some information about from the pharmaceutical sciences side about vaccines basics. Dr. Spitali.
Good afternoon, everyone. Thank you so much for joining us. So I'll be walking through just a few slides to talk about what it means to get a vaccine, how vaccines can be designed, and what the current approved vaccines are and how they work. So next slide, please. So some people might ask, how do vaccines work? How do they get their efficacy profile? And what is the mechanism of that broadly defined? So the way the vaccines work are, we have an understanding of the infectious agent. It can be a virus in this case. And we have an understanding of its mechanism of infection and what happens to cells when a cell becomes infected. We can then use that to, in some cases, design variants of the virus that are weak, or as in the case of messenger RNA vaccines, introduce a piece of the virus into our cells. Our body recognizes that as something that's foreign, and in order to recruit the immune system, it starts to create things called antibodies. And those antibodies bridge a cell that is infected or an infectious agent with an immune cell to try to clear out that infectious agent. And in some cases, you can have this antibody response to defend against a disease for the lifetime of a patient. In some cases, that's not true, but in some cases, you can have this antibody response for the lifetime of a patient. So next slide. So some of the current types of vaccines that are under development for SARS-CoV-2 are built around a basic understanding of the molecular makeup of the virus, which is shown in the middle of this slide. The virus contains genetic material, which can be RNA in this case. It has parts of its capsid, and it also has proteins that are on the outside that are important for telling the SARS-CoV-2 virus which cell to bind to and infect. So in panel A on the top left is an example of a live attenuated virus that can be used as a vaccine. In this case, a piece of a virus can be taken out that's important for this virus to replicate in our cells and spread from one cell to the next. So you can think of it as a weakened form of the virus. In panel B, I'm showing you a whole inactivated virus. This is a virus that's completely dead, but still has some of the molecules that would be recognized by the immune system in order to build an antibody response. In this case, this virus is introduced into the patient and you build an antibody response against that inactivated form. You can use molecular components. So for example, RNA molecules, which I'll talk about on the next slide, which can be added into cells to produce proteins that our immune system would recognize. DNA molecules, which can give rise to messenger RNA, which can then produce proteins. It's just another way of passing the genetic material from the vaccine into the patient to get an antibody response. And proteins, we're going to call these recombinant subunits, which are parts of the viral proteins that are produced and isolated in pure form You can introduce those pure proteins into the patient, and our body would still recognize them as being foreign to build an immune response. Another way to do it is to use a virus that is not SARS-CoV-2, in this case, a safe virus that contains the material of interest that is from the coronavirus that can then be introduced, and this safe virus introduces that material into our cells to build an immune response against it. So there's a lot of ways to design parts of the infectious agent we're interested in, so in this case, SARS-CoV-2, to develop an immune response against it. Next slide. So as Dr. Hirsch mentioned a few slides ago, messenger RNA vaccines are currently being used and administered to patients in an effort to suppress viral infection. In this case, what we're using is a piece of messenger RNA. Remember, messenger RNA is the genetic material that codes for a protein. It is encapsulated into a lipid capsule, just like we have lipids in our body. That lipid capsule is injected into the muscle of your arm, and it goes into a muscle cell because muscle cells are easily accessible through an injection and also highly metabolically active, which means that when the RNA enters that cell, it will undergo messenger RNA translation very rapidly, producing a lot of, in this case, a spike protein. And that spike protein is produced and then secreted out from that cell, that muscle cell, and then our body recognizes it as being foreign and builds up an antibody response to that. In contrast to those other types that I mentioned before, an attenuated virus or a killed virus, this is a pure single molecule that produces a single protein, and that protein is what's recognized by the immune system. In this case, if our immune system builds up a response to that purified protein, the antibodies can stay as part of our immune system to recognize it if you get an infection with a live virus later on. Next slide. So now I want to introduce Dr. Chan, who is the chair of the clinical pharmacy department in the School of Pharmacy and Pharmaceutical Sciences. Thank you very much, and I'll hand it over to Dr. Chan.
Thank you, Dr. Spitelli. So now we've got some background related to COVID vaccine, or at least how vaccines are being produced. What I want to focus on for the next 10 minutes or so is the efficacy and safety of the currently available COVID vaccines. And of course, the focus are the ones that we have available. So next slide, please. So we'll look into the Pfizer as well as the Moderna vaccines. And I'm going to use some data that's being published in scientific literature to let you know about who are the patients actually being enrolled in these studies, to have a better understanding about the safety and efficacy of these vaccines. So if you look across the two studies, right, that have actually looked into Pfizer and Moderna vaccines, you can see there are tens of thousands of Americans from many different sites within the United States are being recruited. And what's encouraging to see is that there's a very wide racial and ethnic distribution of the people who are being enrolled in the study. As you can see here, there's a wide range of Hispanic, African American, Asian, Native Americans, and other ethnic groups included. And what's also important is if you look at the age range of patients who are included in these studies, you can see that there are a substantial amount of elderly patients being incorporated in these studies as well. Next slide, please. So this is basically the nuts of it, right? Is really how efficacious are these vaccines that are being given to the patients who are enrolled in the trials. And if you pay attention to, so to speak, the endpoint, and this is a number that you keep hearing in the media, is that these vaccines are 94 to 95% efficacious. It really means how efficacious it is to prevent an infection of COVID. And here, when we talk about COVID-19 infection, we're talking about a range of mild to moderate to severe infection. So symptomatic infection it is. And clearly, this is very, very good data, because if you look at some other vaccines that are being approved in the market nowadays, efficacy to prevent infection generally ranges between 50 to 60%. So hitting 90% certainly, is a very good sign. But I also wanted to make sure that I let you know about what it means about other concerns that you may have. For instance, does that really cut down the asymptomatic transmission of the virus to other people? Now, at this point, based on the data that we see, we don't know yet. And I think this data eventually will be published. But so we don't really know whether or not that for those people who are vaccinated, is there a chance for them to pass the virus on to somebody else asymptomatically? So which is why it is important to reiterate the message that for those people who vaccinated, they will still need to continue to practice social distancing, continue to wear a mask, avoid crowded places, and some of these other public health advisories. Next slide, please. Another very interesting concern that comes from the media as well as from the public is what's in the vaccines, because a lot of people are thinking that there are other ingredients in the, on top of other things that we don't know about. But I want to use this opportunity to clarify that. Now, in addition to the mRNA, which actually helps to elicit an immune response in our body, there are actually a lot of inactive ingredients that are included in the vaccines. And the purpose of these ingredients, including lipids, salts, sucrose, buffers, and diluent, is really to allow the vaccine, the mRNA itself, to be able to be stabilized when it's being given, and in the process of, that is frozen. And also, of course, to have certain balances in the body in order for the vaccine to work. I wanted to bring this up, the fact that none of these vaccines actually contain eggs, gelatin, latex, preservatives, or adjuvants. Now, it's important because there are people who are allergic to these substances. And if you have an egg allergy or gelatin allergy, that doesn't preclude you getting COVID vaccine. One concern that's coming out is because some people in the very, very small population actually has a allergic reaction to polyethylene glycol. And this is one of the lipid, so to speak, is in the vaccine itself, that may potentially lead to anaphylactic reaction. So if you're aware yourself that you have this particular allergic reaction history, you may need to talk to other healthcare professionals before you actually receive the vaccine. Next slide, please. But I want to reiterate from the trials itself, there's no significant safety concerns being identified in the clinical trials. If you look at the way how the trials are being conducted, patients are being followed up to 8 weeks after they've been being administered with the vaccines. So the vaccine recipients are pretty much followed for about 2 months of period of time. And that's conventional because it's not expected that there will be any types of side effects that are related to the vaccines that will occur beyond 8 weeks. However, I do wanted to mention that there's a high likelihood that for vaccine recipient to have some type of side effect after vaccination. And typically are these non-infectious flu-like symptoms. So fever, muscle ache, headaches, etc. And again, I think that's a good sign because it's essentially your body actually mounting an immune response. Some people may even have pain or itchiness at the injection site, which is also not abnormal. But I want to reiterate the fact that these are not preclusion for people getting the second dose of the vaccine. Because again, for the vaccine, as you probably will find out later on, that there are 2 doses that are being given to patients. After vaccination, patients can also use over-the-counter medications, things like Tylenol, Motrin, Advil to manage symptoms and related to these flu-like symptoms that I'm talking about. And if you have any concerns about these products or so and how to use them, it's good that you talk to a pharmacist, before you buy these products over the counter. Next slide, please. I also want to address that the severe allergic reactions related to these COVID-19 vaccines are available on the market. So within the first two weeks or so, or 3 weeks or so after the vaccines are being given, there are about 5.9 million first doses. There was a summary statistic looking at how many severe allergic reaction, what we call anaphylactic shocks or anaphylaxis, occurred in patients. As you can see here, the numbers are extremely low, very, very rare. If they occur, the average onset is within 7.5 to about 13 minutes. So that also explains why it's important to closely monitor vaccine recipients 15 to 30 minutes after the vaccination. Again, to reiterate the fact that these reactions are extremely rare, but they're also extremely treatable. So that also explains why there are protocols in place where vaccinations are being given. So we need to make sure that there are emergency kits available, EpiPens, making sure ambulance access is available so that, if somebody really has a severe reaction, the person can be well treated. Next slide, please. So the approval of the vaccines, if you look at the current status, both the Pfizer as well as the Moderna vaccine actually have just received the emergency use approval from FDA. Now, this authorization itself, what it really means is that it's being filed earlier on by the manufacturer when efficacy safety data were available, meaning the clinical trial data that I presented to you earlier on. And that data was being reviewed by national and state level committees to ensure that, again, the vaccines that we are planning to give patients are efficacious and safety. But that's really not the end of the approval process of the vaccines. As you can see from my arrow, towards the end of the arrow, in order for the vaccines to get full approval, manufacturers will still need to present 6 months of post-vaccine safety data. So meaning, our vaccine recipients who are getting a vaccine this time, what are safety issues, et cetera, in order for the vaccines to get full approval. So you might be wondering, how is this information being captured, right? And that's the next slide I want to present. Next slide, please. So if you look at what are some of the mechanisms that we have in place nationally in order to capture these adverse events related to the vaccines, we have the Vaccine Adverse Event Reporting System, or the VAER system, that's been in place for a long time. It's a website where people can actually proactively report the adverse events related to the vaccines. In fact, you and I can go to this website and do so. There's also the CSER program, which is the Clinical Immunization System Assessment Program, that CDC works closely with several medical centers to come up with strategies to monitor and to do research on adverse events related to vaccines. Lastly, I do want to say that there's an app called a vSafe, okay? And this is the after vaccination health checker. It's created for the purpose of having an active mechanism for people to for manufacturers and CDC or so to monitor, the symptoms that are related to, the vaccination. So the app itself is programmed that it will actively talk to the recipient to get information about, how you're feeling after the vaccines, et cetera, and have that information collected so that, we can continue to monitor the safety of the vaccines. So, with that in place, we know the fact that the vaccines that we are getting for COVID-19 are safe and reassuring. Next slide, please. So here, I do want to hand off to Dr. Hurley Kim. Dr. Hurley Kim will talk to us about the logistics for the vaccines in terms of administration, distribution, and allocation. Thank you.
Thank you so much, Dr. Chan, and thank you all for joining us today. So we've heard a lot about the exciting science that has gone into creating these vaccines and ensuring that they are safe and effective. But I'm going to talk more about, what's going on to actually get them into people's arms. Next slide, please. So we currently have 2 vaccines available, as I'm sure most of you are aware. There's one created by Pfizer and Moderna. These are the same 2 that Dr. Chan just discussed in terms of their safety and how well they work. Both of them require 2 separate doses. They're administered into the deltoid muscle, just like your standard flu shot and most of the other vaccines that we have. They do require 2 doses for a full immune response. For Pfizer's, those would be separated by at least 21 days. For Moderna, separated by at least 28 days. And I think also really important to keep in mind that these are minimum intervals. If you get your second dose, a week or 2 later than it was intended, that's not a problem in terms of the overall efficacy. I know there's been some concern of what if I don't get an appointment exactly the day that it's due, and that is okay. I also wanted to point out that we are going to have additional vaccines coming soon. So the most likely next candidate will be one from Janssen and Johnson & Johnson that has applied for EUA status and will be reviewed at the end of this month. This one is going to be a little bit of a game changer because it does only require a single dose. So it will be much easier to get this vaccine to patients and get them protected more quickly. Next slide, please. So in terms of storage and distribution, these 2 mRNA vaccines do present some pretty significant challenges with regards to their logistics. So they both do need to be stored frozen. The Pfizer vaccine needs to be stored at what we call ultra cold temperature. So this is either dry ice for a short period of time or in specialized freezers that are usually only found at specific, high-level research institutions. So your average hospital pharmacy, especially in rural areas, is not likely to have access to this specialized type of freezer, and dry ice can only get you so far, especially if it's not being replenished regularly. So you can imagine the challenges for distributing this vaccine, especially in a lot of underserved and rural areas and with decentralized distribution. Fortunately, the Janssen and Johnson & Johnson vaccine helps to solve this problem as well, because it can actually just be stored in your regular standard refrigerator. So something that every pharmacy has, every hospital, and would be much easier for mobile distribution as well. Next slide, please. So the distribution process is quite complicated. There's a lot of steps that come between the federal EUA and the CDC recommendations for the vaccine, and those vaccines actually getting into someone's arm. So there are state and county public health and elected officials, all of which can have an impact on who and how and where these vaccines are administered to patients. We do have a lot of different settings that are currently administering vaccines. So I highlighted there the mass vaccination sites and pharmacies, because I think these are the places where the majority of Americans will end up receiving their doses. And then also should probably add to this mobile vaccination sites that are starting to be in use in some jurisdictions, including here in Southern California. It's really important to keep in mind that the complexity of this process and all of these steps that have to be coordinated in order to get the vaccine to people. But I do feel optimistic that our processes are improving, and as long as our supply continues to increase, we will start to see the rate and the pace of vaccination continue to increase as well. Next slide, please. So there are also a lot of decision-making processes that go into figuring out, who's going to get vaccinated first or next. The factors that are considered here at the most basic level are maximizing benefit and minimizing harm. So this means reducing to the greatest degree that we can the most deaths and reducing hospitalizations. So this is why we prioritize our older adults and also those with high-risk conditions. Also, really importantly, is the focus on equity. So prioritizing groups that have been really hard hit by COVID, I think, is really important, especially because these are oftentimes the same communities that have really helped us to keep society running while a lot of us were at home, working safely from home. So it's really imperative that we do continue this focus on equity. It is not easy to continue to make equitable decisions, but it's really important. And it really is, it's the right thing to do for society in general. If we're allowing virus to spread unabated in our underserved communities, that's where we get these variants that will come back to those of us, even if we're vaccinated, if that spread is allowed to continue long enough. But there are a number of things that are being done in California to help promote this equity-based prioritization. Unfortunately, no single county or city is doing all of these things, but I think a lot of them can be thought of as best practices. So for example, the state of California is using this, what we call the California Healthy Places Index, and this is based on housing, based on access to healthcare and transportation, that tells us neighborhoods and even down to the level of a census tract where there is vulnerability to having a lack of access to healthcare and even having a lower life expectancy. So California is looking at ways to help allocate additional doses of the vaccine to some of these, evidence-based, underserved and disadvantaged areas. Also, things like mobile vaccination. So Long Beach has started to implement some mobile vaccination to be able to get vaccines to people who may not have transportation or internet access, things like that. Also, some jurisdictions are reserving doses for local residents in some of these underserved areas so that you don't see someone from Malibu driving to East LA to get their dose. That should not be happening, although in many cases it is. We're also adding vaccination sites and incentivizing providers to focus on some of these underserved areas. And then the last factor there is just efficiency and minimizing diffuse. So at a certain level, we do need to make this process as quick as possible. And sometimes that will mean vaccinating people a little bit outside of their turn if it means that we don't have to waste a dose. So a dose in any arm is better than a dose down the sink, essentially. Next slide, please. So this is just a very complicated and busy slide, but what I just want to point out is that California is not following exactly along with what the CDC has recommended, and our counties are also doing something a little bit different as well. And it is okay for states and counties to make their own decisions, based on the needs of their populations. There's only 3 states that are following, directly word for word what the CDC has recommended. And those that are highlighted in yellow are the people that we're vaccinating right now. And next in line will be our frontline essential workers. And I think this really speaks to that equity-based prioritization. Next slide, please. So just to give you an idea of who is included in each tier of this rollout. So these are kind of sub-prioritization based on the critical impact that these sectors have to society and also the scarcity of workers. So for example, food workers are included here because they are probably one of the most critical sectors outside of healthcare, and then education included as well, because we don't have a large surplus of people who would be able to step into those slots if we did have a big impact on our education workforce. And just a quick note that education in this context does include our post-secondary educators, so universities, trade schools, et cetera. Next slide, please. So the big question that we always get is, when will it be my turn, or when will my parents be able to get their vaccine, for example? And unfortunately, the answer is, it's very difficult to predict exactly when doses will become available to different groups. So some of the factors here include, how many doses are available. And this is, of course, is going to also be influenced by how many vaccines do we have available? How does prioritization roll out? What's the uptake? So if very few people want to receive these vaccines, it's going to be difficult to we'll move through the phases more quickly, but we'll end up with a lower percentage of our population vaccinated. Also, of course, our efficiency and sometimes just location. So depending on the county that we're in, things may move more quickly or more slowly. And I did just want to kind of highlight that, this is probably the most complex endeavor in human history to try to vaccinate essentially every person on Earth. We are trying to do it quickly. We're also trying to do it equitably and safely and in a way that minimizes suffering. But I kind of think of it like, it's like cooking dinner for 9 billion people and trying to serve them all on time and in a particular order, but we may not know how much food do we have available or how many people are actually going to want to show up and eat. So it's a very complicated process, but public health and in healthcare, we are doing our best. Next slide, please. So just to kind of wrap up, I think it's important to know where to find more information. I also really want to emphasize that it is normal to have questions and concerns and to have hesitation. I didn't know from the very first moment I heard these vaccines were available that it was something that I wanted to get. I went through the process of getting more information and making that decision for myself. And that is a normal process. If you have questions, please reach out to your healthcare providers. The CDC has really great information for the public specifically, and also state and county public health organizations as well. Those websites that we have listed there are for Orange County and Los Angeles County, where you would actually go to sign up for a dose of your vaccine. And that's all I have. I will turn it back to Dr. Hirsch. Thank you so much.
Next slide, please. And thank you to all the panelists for your remarks. And great. Here are some additional resources, and we're going to try to put these in the chat because we know you can't just look at these URLs. So we'll try to do that. Another question I'm getting lots of questions are, can we have these slides? We will be posting the slides. So you'll have this information, and I'll tell you more about that in a bit. But let's turn to questions and answers now. And let's see. So can we get our panelists up? Not sure how you do that. Can you put us? Great. Okay. So we've had a number of questions that came in prior to, and a number of questions, a lot of questions that are coming streaming in here. So I'll just get to it. First, I will say, Dr. Chan, I'm going to try to direct these. Dr. Chan, you mentioned about Tylenol and Advil, but what about in the question is, should patients take Tylenol and or Advil before they get their COVID vaccines?
Yeah, so the current advisory is not to take any of these medications before the vaccination happens. And the rationale for that is because if your body is trying to generate an immune response, by taking Tylenol or Advil, these medications potentially may curb the efficacy of the vaccine by reducing the immune response that you may have in your body. So that's the advisory. Now, even for after vaccination, I would advise that you probably do not want to take any of those medications until you actually have symptoms that you cannot bear. That's because, again, you want to avoid any of those situations. Although I must say, in the scientific data, we don't really have a lot of data to demonstrate that taking these medications actually would curb the efficacy of the COVID vaccine. So I think it's more of a precaution, that we're trying to be, safer, so that to make sure that, we're not going to affect the efficacy of the vaccine.
Great, thank you. Next question, I think Dr. Hurley Kim perhaps can answer this. Let's see. Oh, sorry. So do you know, is there any information on two questions somewhat? We have a question of, should I proceed with getting the vaccine if I have been exposed to COVID? And a related question is, how long should I wait after having had COVID to get vaccinated? Can you comment on those?
Sure. Yeah. So these are really good clinical questions that we have. I get all the time, and I summarize it in this way. So if you have never tested positive for COVID, absolutely you should get vaccinated at the very first opportunity that you have. If you have tested positive for COVID and it was more than 90 days ago, you should absolutely get vaccinated at the first opportunity you have. If your COVID diagnosis was within the last 90 days, there's not a clinical issue with you getting vaccinated. The vaccine will still be effective. It will still be safe. However, we do know that the chance of reinfection within 90 days is extraordinarily low. We don't have evidence yet of a case of that happening. So if you have been diagnosed within the last 90 days, know that there's not a rush for you to be vaccinated, but it is still valuable. If you have an opportunity, I would recommend taking it. Some public health departments are recommending that people with a recent diagnosis wait in order to kind of save that supply. But especially as we get toward the point where supply is no longer an issue, I would say, get vaccinated as soon as you possibly can.
Great, thank you. Dr. Spitali, this one came in earlier and it came in from a couple people. Okay, do you have anything that you can say about what UC Irvine is doing on developing vaccines?
Yeah, so there's currently a few ongoing efforts by UCI researchers that are collaborating with clinical workers to come up with potentially novel vaccine modalities. They are different than the messenger RNA ones you've seen, as we talked about today. They are more focused on using combinations of nanoparticles and proteins that would go into our body and be released to generate an immune response. So those are new avenues for vaccine development that are ongoing at UCI. Very, very different than what's available. And this is more akin, I know Dr. Hurley Kim and Dr. Chan spoke earlier about single dose vaccines. These are more akin to the molecular makeup of a single dose vaccine, as they were talking before.
And I think that points out a good point. We have a number of vaccines coming, and we've got some questions on future vaccines, which we are going to have a follow-up to this with updates. And so we're going to be addressing those later. Maybe Dr. Chan, looking at the data, there's several questions related to, are there long-term effects that we know about of the vaccine?
Yeah, so based on data, we don't really know yet, because again, there's not much data that's pointing out towards that, there are long-term effects of these vaccines. Like I said earlier in the talk, most of the follow-up at this point based on clinical data is up to 8 weeks. But because of all the mechanism and of course all the attention that we have with these vaccines, we are really gathering a lot of safety data. So I'm sure if there's safety signals, we'll be alerted very quickly. So that's good news about a vaccine that's in the spotlight.
Great. Dr. Hurley Kim, can you answer, or maybe Dr. Chan, basically a question a number of people have said is like, why are the side effects more intense for some people after that second dose of either Moderna or the Pfizer vaccine?
Either one of us.
Dr. Chan, you're there. I'm going to let you go.
Yeah. So I think mainly because you are still mounting a response and your body is starting to actually mount a response to the first dose that you had. So when you are exposed again to the vaccine for the second time, certainly your body reacts even harder to whatever that you're getting for the second dose. So that's scientifically, I think it does make a lot of sense that you react so much harder when you're exposed to the second time.
Oh, okay. Dr. Hurley Kim, you mentioned this a little bit in hesitancy. You mentioned that in yourself, but what kinds of advice would you give to people who are either hesitant or either people that are trying to work with patients or others that might be hesitant?
Yeah, so this is an excellent question. I think we haven't talked enough about hesitancy at this point because we've been so limited in terms of the number of doses. But eventually we are going to get to the point where there are plenty of doses available, and we will start to see more of the effect of those people who may be questioning the safety of the vaccine or aren't sure if it's necessary or if it's effective. And my biggest piece of advice is to share information when that person is ready to receive it. So, I mean, you can give them all the resources you want, tell them about clinical studies and things like that. But if they're not ready to hear that information, it's not going to, it's not going to get anywhere. For those who may have questions themselves, I think the most important thing is to find trusted sources of information. So that's people like healthcare providers. It's websites like the CDC. It is not places like Facebook and Instagram who may have a little bit more misinformation circulating. So trusted sources of information are the biggest foundation that we have to talk about hesitancy.
Thank you. Dr. Spitali, can you give us a brief answer, because science is complicated, but the question is, there's a number of questions about what about the variants? And we are getting, information about variants, and that's fine, that happens. But what would be kind of the process from a scientific standpoint of changing or augmenting or altering the vaccines that we have to respond to variants?
Yeah, so I'll break that up into two very brief responses. The first is there's a lot of very promising data that the current vaccines, in which a messenger RNA is producing a spike protein, is still quite effective against the mutant viruses that have been emerging around the world, including here in the US. And in fact, there was a recent paper published in Nature just a couple of days ago detailing how the responses are, in some cases, indistinguishable in their efficacy between the designed spike protein and these emergent variants. That's one thing. The second thing is, a lot of the variants that are emerging are in the protein called the spike protein, which is important for the virus to recognize the cell it's going to infect. Luckily enough, at least in the case of messenger RNA vaccines, the vaccines are designed to take a messenger RNA and have it produce that spike protein and the immune responses against that spike protein. The great thing about that platform is it's very easy conceptually and scalability wise to change the primary sequence of that messenger RNA, give rise to a spike that has that variant in it, and use the same protocol in development, at least on the science side, to test those as alternatives. So I think there's definitely a clear scientific pathway to pivoting very quickly, if necessary, using platforms that are already available for producing vaccines.
That's encouraging. That's good. Dr. Chan, we have some questions about what I would call special populations for the vaccines, given what we know so far, as far as individuals who are pregnant. And another question is children. What do we know about that from the data so far?
Yeah, thanks for that question. And I think so again, these are two questions here. So when the vaccines first came out, obviously people are really trying to figure out if somebody is pregnant or even like, in the midst of getting the vaccine and then they get pregnant, so what happens, right? So first of all, in the trial itself, there were people who were getting the vaccines and eventually got pregnant. And that happens to both of the treatment arm as well as placebo arm as well. And so far, we have not seen any signals that anybody who've gotten the vaccine got into any safety issues, So, again, for some of the data that we have, at least we know that, it's relatively safe. For children, it's a very different story because as you can see from my slide, the tested population of the vaccines are looking at 16 and above for the Pfizer vaccine and 18 and above for the Moderna vaccine. Just very recently that, companies starting to trial on younger patients. So at this time, we don't really know, and there's no recommendation, which is also why we don't see children getting the COVID vaccines. But, again, if you think about the complications that are related to the different populations, seemingly, at least based on the data that we have, with the younger folks, with children, et cetera, the risks of getting complications are much lower comparing to certainly much older populations, like in the elderly. So I think it does make sense that, when we first, roll out the vaccines, it makes a lot of sense to trial on those patients who are older and certainly those people who have more complications, and which are people probably higher priority to get the vaccines as well.
Thank you. Dr. Spitali, this is right up your alley. And as I mentioned in your introduction, you actually are an expert in mRNA research before vaccines ever came around. The question we've gotten several different ways is, we talk about this technology being so new. Can you explain how long this mRNA technology has been in the works? Can you give us a little bit of background that is not so new?
Yeah. So I think this is one of the most important things that society should try to understand on a much deeper level. Messenger RNAs as ways to deliver proteins into cells is a technology that is several decades old now. And the beautiful part about that is there's a very deep understanding of how to get messenger RNA molecules into cells, what they do when they are in cells, what kind of molecules in cells they interact with. And the companies that have risen to the challenge, so BioNTech and Moderna, have been studying messenger RNA delivery for a long time, and through other programs have gotten a deep understanding of the safety and efficacy of these platforms in patients. Now, the real cool aspect of that is because they go through a single developmental program. So there's a target, the messenger RNA, that target, how to package it, how to deliver it, how to study the safety and efficacy. That's the reason that these programs have been able to pivot from their current research focus in those companies to viruses so quickly, is because the platforms have been around for a long time, are very mature. They have a deep understanding of potential safety issues. And so, I think that we have to understand that these things are really quite mature. And that's the reason why it happened so quick.
Great, thank you. Thank you for that. We've got several questions kind of about the new J&J/Janssen, same company, larger company, smaller piece, vaccine that it's coming. And I'm going to ask Dr. Hurley Kim, although I think all of you could answer this. Can you just kind of give what we know now, because we've just gotten maybe a basic look at the data. This has not gone to the FDA yet, but can you just summarize maybe in comparison what you were talking about, the other two, the Moderna and the Pfizer? You had a nice graph there. What do we know at this point in time? And this is something we'll update more when we do our next one of these.
Sure. So what we know about the new vaccine at this point is that it will be much easier to distribute, to administer, also that it is highly effective. So I think a lot of the headlines, might make us think that 72% of efficacy is not as good as 95% efficacy. But some things to keep in mind are that none of these vaccines have been in head-to-head trials, meaning we've never had a trial that directly compared Pfizer versus J&J in the same patient populations. The J&J vaccine was also studied later on when we did have some more of these variants in circulation. And also, I think probably the most important take home when we talk about efficacy is that for all of the vaccines that are considered to be candidates or are available, they have all had 100% efficacy against very severe disease, meaning requiring hospitalization and also death. So, there might be slightly higher incidence of moderate or mild symptoms in different vaccines, although we don't know that for sure. But at the end of the day, all of them are very effective at that most important endpoint.
Great, thank you. I'm going to punt this over to Dr. Spitali, though you guys might also want to comment. Does having a strong immune response, like having flu symptoms, having these symptoms after your second vaccine, mean that you have a stronger or weaker immune system?
Gosh, I don't know. I don't know the exact answer to that. But as Alex mentioned earlier, Dr. Chan mentioned earlier, when you have your second dose, your immune system is reacting, and your ability to create a fever, have soreness, have fatigue, those are all measurements that your body is springing into action. And those are good signs. I think oftentimes we say, oh, I had a strong reaction to my second dose of the vaccine as a negative. Well, it is negative because we feel that way, but actually our body is responding quite beautifully to and doing what it's supposed to be doing. But in terms of if that means you have a strong or weak immune system, I don't know the answer to that.
Dr. Chan, any thoughts on the end part of that?
No, but I do want to emphasize, because again, people are worried that, because they're not getting, some sort of response after they receive a vaccine, meaning, it's almost like comparing symptoms across friends. And the truth is, I think everyone is individualized and different, right? So you might be having a stronger response versus another person, but it doesn't necessarily mean that, you are not getting the efficacy of the vaccine. So I think that is important to think about.
And I think just from what we know about vaccines in general, side effects are highly variable, but efficacy does not tend to be highly variable. So I would not suspect that, a lack of side effects means that it didn't work well, or that if you had a lot of side effects, that means it did work well. These vaccines also cause, extraordinarily high levels of antibodies, much higher than the average infected patient. So the side effect profile, I would not say is predictive of how well it worked or didn't work.
So Dr. Hurley Kim, I'm going to stay on you for this one, and maybe Dr. Chan can chime in too. A number of questions about, okay, I have allergies of some sort, whether it's very different kinds of allergies. What advice would you give patients that have various allergies?
So I would say it depends on, what is your reaction? That's always my first question when somebody says they have an allergy to this antibiotic or to something else. What happened when you took that antibiotic? If it was something like a serious rash, of course, if you had something like anaphylaxis, we will ask you to wait for 30 minutes instead of 15 minutes after your vaccination, just in case something is to happen. But there's not any particular allergy that would preclude someone from being a candidate for vaccination. Just means that we would need to be a little bit more careful with observing them afterward.
Dr. Chan, you want to comment?
Yeah, I echo Dr. Hurley Kim's comment as well. If you look at those people ended up having severe allergic reactions at least from the documents that we've seen, the data that's being captured, a good number of them actually had a history of severe allergic reactions as well to some sort of drugs or some sort of substance. Now, when I said a severe allergic reaction, I'm talking about people who are having shortness of breath, ended up needing to be hospitalized, very, very severe reactions, that is. So I think in those situations, I think it's safe to be monitoring more closely in those people, and also making sure that, after getting vaccination, these people are being monitored. To me, I think that is the key message here.
And you have been involved in organizing the pharmacy service at our Bren Center, we're at UCI, where we are doing some vaccinations. What's the experience been there, if you can say, as far as adverse reactions?
Yeah, so we are 5 weeks in business now at Bren Center. Very proud of the fact that we are able to serve the community. One thing that I'm also very happy about is that we haven't really utilized our ambulance or EMS services, for people who are having severe reactions, etc. In fact, we haven't seen any. So, knock on wood, it's great news.
Yeah, that's great. Okay, we are nearing the end of our time. We have 3 minutes left. I want to first thank our panelists. We'll give you you can't see everybody else clapping, but I'll clap for them. We appreciate you taking your time. So let me tell the audience what will happen next. We will be having an encore, what we're calling an encore session, but it's probably going to be more of an update session. And that will be on March 1st. And you will receive announcements from that. There are a lot of questions in the Q&A we didn't get to. I think a lot of them we might repeat, and we will be able to give more information next time. And will certainly be more updates on the J&J vaccine for our next session. We will post the slides on our website, so you can see those, and those will be up fairly soon. And until our March 1st, I really wanted to thank everybody, for joining us and for the great list of questions. There's like way too many questions than we can answer right now, but we will continue our conversation here. So in the meantime, and several of the speakers said this, please make sure you maintain your social distances. Even though we have vaccines, you wear your mask, wash your hands, and avoid large gatherings, and keep us all safe. So until March 1st, we look forward to seeing you all again. Thank you. Thanks for attending.